So I'm really happy to introduce our local talent, Stefan the Folter. He's gonna talk to us about functional genomic, flower development and fruit development. Stefan studied plant biotechnology in the Banghal Larenstein University of Applied Sciences, and then did his PhD at the Radboud University with Gerko as his advisor. Then he was a junior researcher in the Plant Research International Institute at Wageningen in the Netherlands, and finally he became a full professor at Langevio Simbestad. So he's currently SNI 3 and the current director of the graduate program at the Plant Biotechnology at Simbestad. So as you know, he has a long list of publications and he has made important contributions in plant development genetics, particularly identifying regulators controlling the formation of tissues that make up the flower in Arabidopsis. So thank you, Stefan, and please go ahead.
Thank you very much.
Now? Yes. Okay, thank you very much for the invitation and for the introduction, Carlos and Alfredo Herrera. Sorry for the difficult names that you had to pronounce. So I'm Dutch, so I studied in Wageningen, so I was there more or less 10 years before coming to Mexico, to Langevio. So now I'm more or less here 15 years. And I would like to talk a little bit about what we are doing related to fruit and flower development. And especially to, okay. It doesn't want to move. Now, especially as today that we talk about food, so it's, I think, nice to talk about where does our food come from. Yeah, perfect. Thank you. Now, and to remind you, and well, it's below there, under the bar of, now with about 80% of our food that we eat normally daily comes from flower and fruits. Now, so this is, well, understatement to say it's very important. And what we really like to understand is the biology now. So plant biology, which genes do you need, which factors, hormones, for instance, are needed, are guiding these developmental processes. So we study different species, but a lot we study Arabidopsis. And to show a little bit here, so this is a small plant. So when it flowers, it forms an inflorescence, and there at the top it has a meristem, and it forms different floral buds in a spiral. Now, in each floral bud, in the end, produce a pistil or a gynoecium, which in the end turns into a fruit. So there are many mutants known. So at the end of the 80s, the first transcription factors were known that are really guiding the basic processes of flowers. So here we see all, well, I hope you can see classical mutants, floral mutants, where we miss certain floral organs. Now, and this is because one transcription factor doesn't work, and normally they turn on all developmental pathways. Now they do this mostly together, so they can form interactions with other proteins, other transcription factors. And during the talk, I will come a little bit back to that. Now, so our simple question basically is saying, so how a floral bud produces this structure, this gynoecium. And this is not to read, but just to give an overview that nowadays we know many genes, many transcription factors. So here we see different developmental stages. So the process from a floral bud to getting a gynoecium is more or less 1 week. And here we see just a few days, let's say, of this developmental process. And we see many genes that nowadays, well, there's knowledge from many different labs in the world, also some of our own lab, that we know how these transcription factors work, how they connect to other genes, and different hormonal pathways. And so that was, let's say, the early development, and this is the later development. So when we talk about fruits, also there are a lot of genes are known. So we talk about this structure. This is the mature gynoecium, and this will turn into, let's say, into the fruit. Now we see here just a part of it with seeds. And also here there are many transcription factors that really determine all these very few, well, very fine cell layers that can be bigger cell layers or very fine cell layers that are needed, for instance, that seeds can disperse. So what we do, we use different techniques. This is just different images here. Well, things that we studied in the lab, let's say different genes we clone. So basically we use a lot of genetics, forward genetics, reverse genetics, transcriptomics, computational biology. We do a lot of screens in yeast or other, also in plant systems to identify interactions between transcription factors, because that's very important. And in the end, also about protein localizations, proteins in the plant, and also about hormones. So I will also talk about hormones during the talk. And this is to show some examples. So I'm not going into gene names. Now, but the point here is that too less gene expression or too much expression of a gene can alter developmental process really a lot. No? And this is basically actually also how traits are selected during domestications. At the end, it comes down to changes, mostly comes down to change in gene expression, which can be more or it can be less. So on the left side, we have examples when there's more gene expression. And on the right side, we have, let's say, mutants where the genes are not working, so we have less expression. Now, so basically in this orange block, so we find genes that are really needed for fruit formation. And also when you don't have them, for instance, you don't have a fruit. So here we see a flower basically which is empty in the center of fruits that are developing, although they can be smaller, but they are, for instance, they don't produce seeds. So parthenocarpic fruits is very interested. So seed production, obviously, and also shape of fruits and size. For instance, here we have one that really grows very different than normally it should grow. And the other part is, besides the transcription factors on the other slide, is hormones. So also not to really go in detail. So hormones are very important. So just to mention two is auxin and cytokinin. So auxin is more for differentiation, so to get an organ, and cytokinin is to maintain meristematic activity. So this is a hormone that you need for cell division, let's say. And this is very active in meristematic tissues, which I will come back a little bit to it. So here we have an image. I assume here for you it's very small, so later on we see it a little bit more. So where we have high cytokinin signaling, which is very important to get the different structures in the gynoecium. Now and afterwards to get the fruit. So we'll focus more on cytokinin. So what has been discovered some years ago, first in rice. So there was a mutant where an enzyme that degrades cytokinin doesn't work. So actually you have more cytokinin. So when you have more cytokinin, you have more meristematic activity. And this results in more yield. So you will get actually more flowers. More flowers means more fruits in the end. No? So this is a very nice example, a very basic science and a very key hormonal pathway. When it doesn't work well, you can get more yield. So this has been shown later in Arabidopsis. So we have been studying cytokinin signaling now for quite some years, over 10 years. So one thing is so we do a lot of imaging in using a confocal microscopy. So here we see a marker that is marking the transcriptional activity of cytokinin. And it marks actually a part of the gynoecium. So this is a transverse section where there is the meristem. And there you see very high activity of cytokinin signaling. So this is needed for cell division. Now, again, to get the different structures. So when we have too much signaling, we can get this type of structure. So when we apply cytokinin exogenously to the plant, we get this overproliferation. So here we see in SEM, these structures actually coming out of the gynoecium. So showing that so the balance of this hormonal pathway is very important. So you should have enough signaling, but also not too much. And one structure, so here we have artificially colored the gynoecium. So this is a transverse section. So we cut it, let's say, there in half. So I just want to mention one structure, which is called a rayplum. So with this red bar. So that's actually this tissue, which is in between the two valves of the gynoecium. Now, so Arabidopsis has a gynoecium that we can compare to beans, no? For instance. So there are two halves. When the fruit is dry, it opens, and the seeds will fall out. Now, so how easy this at the end the fruit can open depends on the structure. So the structure that is there. So the width of that structure determines the easiness of opening. So when it's wider, it's more difficult to open. So this, maybe, I hope you can see this. So also here cytokinin signaling is very important. So when we use transactivation constructs in Arabidopsis plants, we can change endogenous concentration of cytokinin. So we can lower the concentration or we can increase the concentration. So when we increase the concentration, this rayplum gets much wider. And when we decrease it, it gets less width. So this is showing that with that cytokinin signaling is needed for this structure. So here are also these type of images, so all SEM images. So we are looking to the width of this structure now. So actually there's a mutant in rapeseed, in canola. So it's very typical for this species that the fruit, when it's dry, it opens very quick. So a lot of seeds are lost actually in the field. And it has a natural mutation in one of these genes. And in Arabidopsis also this exists. So we see here a dark line, and actually it means that this tissue almost doesn't exist. So just the two valves are directly connected. That's why we see this line and not these cell files in between. So this actually we can rescue by applying cytokinin. So meaning cytokinin signaling is below these transcription factors. And so this is a way actually to, or this could be an application actually in a crop, for instance in canola, to overcome seed loss. No? So one other thing, so cytokinin, so hormones are sensed by receptors. So these receptors should be expressed in the cell, and let's say the molecule, the hormone, will bind to these receptors. So in Arabidopsis, so you have 3 receptors. So this is the expression pattern of these receptors. So it's not so important. They are not exactly the same, but they are highly expressed in the gynoecium. And so when we have different mutants, single mutants or double mutants, we can measure and we can observe what is happening in the gynoecium. And I'm not sure if you can read, I think the text is very small. So this is ovule number. Now, so the ovule number, when they get fertilized, they convert into seeds. Now, so when, for instance, here is one double mutant of the receptors, the ovule number is really less. So this mutant will produce less seeds. And also this is again showing that cytokinin signaling is actually very important for yield. But I think this, skip that part. So that were all early functions of cytokinin. So there are also some later functions. So in in crops or in species, even in Arabidopsis, although it's not really a crop. Now we call this pod shattering when the fruit opens. So the fruit, or the xylem, we can call it, when it dries, it breaks open and the seed comes out. So different mutants actually exist where the fruit doesn't open. So they miss a certain structure. So they are all transcription factors. So the names you cannot really read, but so one is called indehiscent, so it doesn't open. Another one, genes are called shatterproof, so also the fruit doesn't open. So they miss certain structures. And there's another one, a mutant, which is called fruitful, which actually is the opposite. And so here we are looking to the cytokinin marker also to see if the signaling is present or not. So the point is that these mutants where the fruits don't open, they are lacking this late cytokinin signaling. And so where the arrows are, so these are these two mutants that don't open. So when we apply cytokinin to these fruits, actually the structures, they do develop. So also here's an example that cytokinin signaling is below these genes, and we can rescue these type of mutants in the fruits, they will open. So also here's a very, let's say, interesting phenomena that we found, and also this in a way could be translated to crops. So here we can see it in real life. So the fruits, they don't open, and when cytokinin is applied, these structures again develop and these fruits do open. So we don't use only Arabidopsis. So this is the example of canola that I just mentioned. It has fruits that open very easy. So here we can see this structure, what I just mentioned, the rhipium, that it doesn't exist in canola. When you apply cytokinin, it develops again this structure. And these fruits actually open less easy. And also you have this ectopic proliferation when we apply a lot of cytokinin. And actually one thing also here related to yield is, so when there's more cytokinin, there's more meristematic activity, and you end up with many more ovules. Well, and actually they do not really fit anymore, so they are just bulging up there. And so what we see is that the yield really increases when we count seeds in the end. So also the yield is increasing. One interesting thing that we found is, so when you look to when we evaluate the progeny, so we don't apply cytokinins, but still the progeny actually has the effect of receiving the cytokinin in the first, that's in the parent. So we still see an increasement of seeds. Also, probably this is something, an epigenetic effect caused or induced by cytokinin. So one thing is we really like to understand genetic networks or small circuits, where we connect the different genes. And you have an example of different transcription factors that are connected to a hormonal pathway. And actually, they are connecting two hormonal pathways. One is cytokinin, and the other one is auxin production and auxin flux. So I will talk a few words about this transcription factor, which is called SPATULA. And this transcription factor, this mutant, has been known for quite some time. So the mutant is showing or presenting this type of fruits. So they are very short and they have very few seeds. And this has to do with that many things in early developmental processes in the gynoecium are going wrong. For instance, there are very few ovules, and this is the result. So to go fast, so this is the expression of this transcription factor in the gynoecium, and this is very similar to the expression of the cytokinin response line. So there we started to think that there could be a connection with this transcription factor and cytokinin signaling. So this is, well, first of one of the, let's say, the connections. So at the end of the cytokinin pathway, the transcription factor SPATULA is very crucial. Now, so one of the experiments that we do, for instance, is looking at the cytokinin marker in the mutant background of this transcription factor. So we lose the expression of this marker, so meaning you need the gene. And this is the opposite. When you have more of the gene, you see much more expression. Also, this is just one of the experiments that we did. And another one is applying cytokinin. So normally in wild type, you get this ectopic proliferation. And this response is different in the SPATULA mutant. And also when we take some genes from the cytokinin signaling pathway, also this response is missing actually. So in a way explaining why you get this type of tissue when you apply cytokinin. Now, so we can, let's say if we go back to the tissue, so the different cell types, we can overlay our network explaining how the development is working and how you are getting the different tissues at early gynoecium development. That at the end affects seed yield and fruit development. And just jumping a little bit, so we also started with tomato. So here we, just to present an example. So for instance, this transcription factor SPATULA, does it exist in tomato? Yes, it does exist. And we are looking now to if the function is conserved. So what we are doing is using CRISPR-Cas editing, using tissue culture, mutating the gene and see what is really happening in the plant. So in tomato, there are two genes. So the SPATULA and there's a paralogous gene, very close. So we created different mutants or different mutations or changes in amino acids, or for instance, stop codons, and also where we have truncated proteins. And then, so here's the phylogenetic tree, so the genes. So here's just an example that we recently obtained, and I hope you can see. So this is a wild type tomato. So this is in microtom. So the title you cannot exactly see. But so here are the mutants. So I hope you can see. I guess it could be difficult. So there are all kind of, let's say, spickles on the peel of the tomato. So they are lighter colored, and also in the green you can see other colors of green. So we think there's something wrong with flavonoids or carotenoids. And now with Jose Ordaz of Lancabio, we are doing metabolomics on these tomatoes to see what is happening. Now, so this fruit, of course, is very different than Arabidopsis. And other type of genes that are very important in Arabidopsis, many times what has been seen is actually they are involved in ripening in tomato. Also here, this gene actually seems to has to do something with flavonoids or carotenoids. So not exactly the same. So it seems like it has some new functions in tomato. And one thing, so I mentioned about protein-protein interactions. So this we can do, for instance, in yeast. So we express two proteins in yeast, and then a system that is called yeast-2 hybrid. When the yeast grows, it means there's interactions between the two proteins. So this is a way how to screen for interactions. So one thing is to see if a protein has new functions or maintained the same function, is to do heterologous 2 hybrid. So taking the protein of tomato and try the interactions with the ones of Arabidopsis. So there are many gene names there, so the all transcription factors very important for gynoecium development. So the names are not important now, but so here we have SPATULA from Arabidopsis, and here we have SPATULA from tomato. And the point that you can see is, so when we see yeast growth, there's interaction. So here are many interactions, and here we almost don't see interactions. So this shows that this protein, let's say on the biochemical level, on the way how it forms interactions in yeast, we suppose it's the same in plants. No, is very different. So this is also a way to see that the function of this protein is not exactly the same in Arabidopsis compared to tomato. And doing also this protein-protein interactions, we also did this in Arabidopsis. So now we are shifting back. So what we did is, because they are so important, these interactions, we cloned all the transcription factors that we know are important for gynoecium development. So they are more or less 80 transcription factors, and we tried each combination, the 80 against the 80 in a matrix way. So here are 80 names, and here are the other 80 names. Now, so each square in blue, it means there's an interaction. And we can translate this in this type of networks. So each circle is one transcription factor, and each line means one interaction. So there are many interactions. So now we can understand better how a transcription factor does it function, which other transcription factor it works, or which other, it can be more than two. And this more than two, if it's a dimer or it's a complex. So what we did is, so these are all the stages of gynoecium development. And yeah, you cannot read anything of this. So there are many colors and many small squares with networks. And the point is, what we did is, so we took the different data, so protein-protein interaction data that we generated. We took expression data that was available, and we took genetic data that is available. So what is the phenotype when you mutate the transcription factor? So by combining all that data and thinking or looking in time how the tissues are developing. So this is basically the meristem. And then during time, so this is, let's say, the time of a week. At the end of the week, we have all these different tissues of a mature gynoecium. I mentioned before, for instance, a rayplum, or you have ovules. So you can follow this line. So from the meristem, where many genes are expressed and all the interactions we are assuming are taking place, during the time we can follow this line, and basically these networks become smaller. Because the expression of these genes are changing. Different genes are expressed only certain stages, and also the function that we know is, for instance, these genes are involved in ovules, and when we follow the other part, for instance, there are genes that are involved in valve development or in rayplum development. So these are now predictions of smaller networks. It could be one big complex. They could be various different complexes. And this is for future work now also to confirm this really in plants if they are really formed. And so I don't know how I go over the time, so a few more slides. So also, if we look again to SPATULA and we look towards interactors, there's a small network. So this is based, okay, a few minutes. I go faster. So this is based on yeast. So as mentioned, we're also looking now in plant systems. So in plants, there's obviously an advantage over yeast because, first of all, the transcription factors are from plants, but also the localization is very interesting to see. Now, so transcription factors normally actually are in the nucleus, but we see if we test interactions and we see where they are localized in the cell. So this is an example where they are in the nucleus, but they are not always in the nucleus. So for instance, here we have an interaction that is in the cytoplasm. When we change combinations, we can move from one localization pattern to the other one. And we can also do this in protoplast, and now we are starting to do this in complete plants using native promoters to express the transcription factors and really see during development how the interactions are taking place and how they are changing. And also the dynamics of these interactions. And we're also starting with IP immunoprecipitation of these type of plants to see which of the proteins are interacting. And so finishing, so I mentioned before, so we use publicly available expression data in using and predicting our networks. So the data is very nice, but still it's normally from the complete inflorescence, for instance. There's a big mix of cells that was used. So one thing is to isolate actually cell type specific transcriptomes to get much more finer expression data about transcription factors. So this we did, so we can do many comparisons. And this is a way also to find really new markers. And these are all transcription factors that we know. And yes, we can, as expected, we can see exactly in very specific locations of the gynoecium, we can find them. And the interesting thing is we can find, of course, new genes that are expressed in specific places. So here we, for instance, we took one gene that's expressed in this tissue. So here's this meristematic tissue and the septum will be formed. So when we mutate that gene, now actually inside the septum is not really formed anymore. So this is an example of this strategy also works for finding new genes and new candidates for gynoecium development. So with that, I like to end. So I hope I could show you a little bit of focusing on cytokinin. So an important hormone that has effect on meristem activity, also carbon initiation. So I didn't mention much about that today. Gynoecium patterning, pod shattering, the opening of fruits, fruit size, and at the end also yield. So with that, I like to end. So I thank many people, the current lab and all the past members of the lab, our collaborations and our funding sources. So with that, I like to end. So thank you very much for your attention, and I'm happy to take any questions. Thank you.
Thank you, Stefan, for a great talk. We have time for one quick question.
Thank you. I got two. Thanks for the study, but they're quick. The first one is how conserved are these flowering pathways in different groups of plants, and the second is if you done gene editing of any of these pathways.
So they are very conserved in plants. So many of the transcription factors, and also let's say the classical flower mutants that I showed in the beginning, those genes are really very conserved. So also in general, let's say the 80 transcription factors that I mentioned that we tested in the interaction screens, most of them are conserved. So sometimes it's, let's say some of them you cannot find exactly the ortholog, but you can find homologous genes. But most are really conserved. About the editing, we just started editing once, or I don't know. So we are not going for all these genes, but in general, there are labs that are studying different genes, different transcription factors in different species with editing. Thank you.
Very much. Let's thank Stefan again.
Thank you.